Today is, as I said, the inaugural Richard Norris lecture, and this is an annual event that's beginning this year. And I'd like to begin this by inviting Professor Dave Chokino, who's the Director of the Institute for Applied Ecology here at the University of Canberra, to take you through a little bit of the background behind this lecture and what we hope to achieve with it in coming years. So I'll pass over to Dave.
My main duty is to welcome you all here to this inaugural Richard Norris lecture. As Ross indicated, we're launching this as an annual lecture, and it's to both recognise and acknowledge Richard's enormous contribution to the field that all of you people are involved in. When Ross first suggested the concept of this lecture, I was struck by how timely and how significant doing something like this would be. One of the first tasks I walked into when I took the role of director on 18, 19 months ago was the need for us to recruit a new professor in water science for the Institute to replace Richard, who had passed on by then. After Richard retired from the role of Professor of Freshwater Ecology, the fort at the Institute was held to some degree by today's speaker, Trefor Reynoldson, and I spoke at quite a bit of length to Trefor about the sort of person we'd be looking for to fill the role or the gap that had been left on Richard's departure. And Trefor always came back to the fact that in addition to very, very high levels of science credibility, a successful candidate to fill the role would also need to have a clear focus on science as a central part of policy and management. And that was really the brief that we went out to address. I think we were pretty successful in achieving that with Ross's appointment, and that really signalled the start of a re-energising process for water science at the Institute. Since then, we've recruited a number of new staff. Ben Kefford is here, I think today. I saw Ben at the back there. G'day, Ben, as an assistant professor, and we have 3 new professors starting at the beginning of 2014. This lecture is a sister event to the Krebs lecture, which we've been running for a number of years now, and that lecture occurs in February. So, if you like, we're looking at the 2 lectures as bookending the summer and giving a taste of the Institute to people who would like to come and hear about the things that we're doing. What I'd like to do now is hand back to Ross so that he can introduce Trefor as the inaugural lecturer in this series. Thanks.
It does give me great pleasure to introduce Trefor Reynoldson as the inaugural Richard Norris lecture speaker. I met Trefor, we've met before, but really only in passing earlier today, and it gave me great pleasure to say thanks, Trefor, for leaving the group in such fantastic condition so that when I arrived, my job was so much easier. Trefor stepped into the Institute at a really difficult time after the loss of Richard, and he took the Institute's water group to new and exciting places, and it's been really a fantastic experience for me to move into the group and see that it really was so successful even in that difficult time. So Trefor received his PhD from the University of Lancaster in the UK in 1983 and his MSc in aquatic ecology from the University of Calgary in 1974. He then worked for the National Water Research Institute, which is Environment Canada's aquatic group, from 1987 until his retirement in 2004. And over that time, he really established himself as one of the preeminent aquatic ecologists in the world, particularly in the space of bioassessment and biomonitoring. He is now a senior scientist at Ghost Environmental Consulting and an adjunct professor at Acacia University in Nova Scotia. His research has been looking at developing improved techniques for bioassessment using benthic invertebrate assemblages, and he has most notably in recent years been associated with formalising the reference condition approach as the standard national biomonitoring network for Canada. In addition to all of those measures of impact and success, He has also been involved in over 150 publications and reports, more than 90 of those in the primary literature, and his expertise really lies in the application of multivariate data towards applied outcomes, particularly in the bioassessment and biomonitoring space. So as I said, it really does give me great pleasure to invite the inaugural Richard Norris lecturer to the stage, Professor Trefor Reynoldson.
My talk's going to be about predictive modeling. And I always liked history a fair bit, so I think it's kind of important that you know where you came from. Maybe not this far back, but this is a problem that's been around for a long time in terms of deterioration of water quality. So before you go forward, it's good to go back. I was at a meeting in the Yukon actually at one point, and some young lady who had just started with Environment Canada said, well, there's this kind of reference condition approach. Some guy called Reynoldson did some work, and I was just sitting there chuckling away to myself. So measuring aquatic systems, this can be done at a whole array of scales, from the molecular to the community. The characteristics of these different scales are different. So at the sort of very sort of fine biological scale, those tests, and we were talking about this morning, tend to be their functional responses at organism and suborganism level tend to be very specific to responses, although we had some debate about that. I kind of think they are, others think maybe they're not. But they're not very integrative, and they have low ecological relevance. They have a rapid response time. And then at the other end of the spectrum are population and community, and we work largely at the community level, and that's what I'm going to be talking about, that community level. These have high ecological relevance, but the response times and the delays are considerable. This is a slide that has been sort of shown various times. The business of bioassessment with degrees of scientific rigor have been around for quite a long time. So classically, the first one was the Saprobic system developed in Germany in the 1890s, early 1900s. The pale area kind of encompasses my career. So when I started in the 1970s, we were very much working with diversity indices at that sort of level, and gradient study designs along rivers. You just took samples along a river, took 5 replicates, measured the species diversity, taxonomic richness, and kinds of things. And then in the 80s in the UK is where this work was pioneered, and Ewan here is kind of the latest incarnation of the RivPax group in the UK. Developed this, what we thought was brilliant system of predictive modeling. And and Chuck in the US, myself in Canada, other colleagues in Europe, and of course Richard Norris here pioneered these approaches in their relevant countries. So I'm talking about sort of this kind of area in terms of bioassessment. This is my timeline. So when I started, a book by Noel Hines, written in the 60s, I think. Is that right, Chuck? About 1960. Thank you, Ian. 1960 was really still, and I think you'd say, the Bible in terms of how freshwater invertebrates responded to pollution. And in that period, sort of diversity indices were the thing. They were the latest thing, like functional traits are, they're the kind of in thing now. So diversity indices were the in thing at that time. I was trying to dig out a copy of one of my first reports. Actually, I'm kind of glad I couldn't find it, because it would be somewhat abysmal. If you remember, I think Simon, it was you when Richard received his award, got Richard's old thesis out, and beautifully embarrassed him, I think, but it's very appropriate. The work you do in your early days, you probably don't want other people to see it later on in your careers. In that time when I first started, multivariate stats were first becoming available. There was a mainframe computer at the University of Alberta, which I was able to program. 2 days later, you get your outputs back on a big spreadsheet, a multivariate PCA plot or something. Then in 1984, that's when John Wright and Mike Furs and Dorian Moss and Patrick Armitage, I think those are the authors, published the first RivPax paper, where they linked habitat to community structure, community assemblage of invertebrates. And pretty much talking about invertebrates exclusively in this talk, that showed that you can in fact predict what you should expect to find at a site if it's undisturbed. So this is the basis of predictive modeling. That RIVPAX program has gone through 4 iterations, Ewan, and has now been morphed into the the Water Framework Directive in Europe. About that time, in that time, in the 90s, coincidentally, both Richard and I started working on these approaches in our relative countries. And we met at a workshop in Canada that I was able to hold in 1990, I think it was. Interestingly, Richard was able to get Osrivas up and running within 7 years as a national program. The Canadian equivalent didn't get going in Canada until about 2000. It took us almost 15 years to get the thing going in Canada. And the reasons for that are interesting and relate to policy and the link between science and policy and management. But there was this period, I think in the 90s, which I look back as the golden age in terms of predictive modeling. I don't know whether you feel that, Chuck. But it is the golden age. There were a lot of resources available to do this kind of work. And I think, as you'll see now, that impetus has kind of petered out a little, and we need to possibly ask the questions, why? I know a lot of you in here understand what we mean by predictive modeling, but some of you probably don't. The traditional method for doing impact assessment, bioassessment in streams, up until the development of this reference condition approach by the RIVPAX people, was if you had a stream, you sampled upstream of the putative discharge or disturbance, and sampled downstream, and looked at how the stream had changed along that gradient. That was the traditional way of doing it. What this reference condition approach, or the RIVPAX approach, is you don't have a single control site. What you do is, in your study area, and it can be defined by a geographic boundary, a watershed boundary, a political boundary, but within that geographic confine, you assign an array of sites that will capture the range in normal biological variation at reference sites. And these reference sites, in an ideal world, and in some parts of Canada it is ideal, these are essentially pristine sites. But in the UK, clearly they're not. I mean, it's been, since the Ice Age, there have been people living there, and it was one of the first industrial countries. It's where the Industrial Revolution began. Very dense population, a very small area. So reference sites are not necessarily pristine. They're the best achievable, they're the best you have available, and you need to be very specific about what they are. But people tend to get reference and pristine mixed up sometimes. So what you do is sample an array of sites in what you consider to be reference areas. So in this little figure, reference sites would not be assigned where there was a lot of logging activity or a lot of agricultural activity. But you want to capture as much as the normal variation as possible. So you have small, steep streams. You might have lowland, wide streams, slow flowing. All those can be reference streams, and you capture that biological variability, because those are going to define your reference condition, and that's what you are going to be comparing any new site against. So the process in building these predictive models, it's really a two-step process. First is to classify those reference sites in terms of their biological community. So we'll say, we have a group of sites here that have similar assemblage of organisms. They seem to be similar to each other. A group of sites here that have a similar assemblage of organisms. They seem to be similar, and so on. And then you match those to your habitat data, so that for a given set of habitat attributes, you can say that given these habitat attributes at my site that I want to see if it's in reference condition or not, I should compare it to this reference community, or this group of reference sites. And this is done using a statistical technique, which we don't need to go into, but what the statistical technique does is it gives you a statistical probability of your test site belonging to one of these assemblages of organisms. So that's what the model does. And then the assessment, there are two approaches to assessment. This is the one that is most commonly used. It's the approach that's used in Australia. So it takes the probability of your site belonging to one of those reference groups, And from that, it does two things. It gives you an expected number of organisms, and it tells you which organisms you should expect with a given probability. And it does it pretty simply. It simply multiplies the probability of a site being in any one of those reference groups by the occurrence of that taxon in the reference group, and you simply sum those probabilities, and you get a combined probability of your taxon appearing. The measure of disturbance is by just adding up the number of expected taxa, by adding up the probabilities, and your observed is the number of those taxa that you find at that site, and that gives you an observed to expected taxa ratio. And in an ideal world, it should be 1, and then you are in a reference condition. So that's the RivPax approach. Richard Norris's comment to Ralph Clark, who I think was Ralph that developed this method, with the other RivPax people. He said, Ralph, that's pretty simple. We can do that. And Ralph Clark's response was, simple, Richard, yes, but elegant, elegant. The advantages of this approach are that it uses all the reference sites, because it's using the weighted probability of a test site belonging to each of the reference groups. And it provides a list of expected taxa. And to me, that's a very powerful tool, that list of expected taxa. And again, we were talking this morning about, going beyond pass and fail, the site's different or not different. This taxa list, in fact, can tell you a lot if it's out of reference about what the potential cause of it not being out of reference is, because different taxa respond in different ways. The disadvantages, and this is a particular case for us in Canada, is the assessment is based on number of taxa only. It only uses richness. There are other attributes. It's still based on presence-absence, I'm presuming, Ewan, in the UK. So changes in abundance are not incorporated in it. And that creates a big problem for us in Canada, because we get what I call a banding bottleneck. Because we have very few numbers of taxa in our streams, where these are cold northern streams. So if we lose 1 or 2 taxa, we hit 0 quite quickly, and there's no sensitivity, or not sensitivity, there's no, what's the word I'm looking for? What? Scope, that's it. There's no scope in the range of the assessment. So we take a different approach in Canada. What we do is we take the group to which the test site has the highest probability of belonging. We plot those reference sites in ordination space. So what you're doing in ordination space is deriving 2 variables that capture all the variation in all the taxa. And it's simply a measure that the closeness of a site together is a measure of how similar it is. So each of these will be a site. And then plot those test sites in that space. So the further a site is away from the cloud of black sites, the more different to reference it is. It incorporates changes in assessment in relative abundance or relative abundance. So we don't have the banding bottleneck that we get with the O to E ratios. The big disadvantage of this method is if your model predicts it to the wrong group, you're comparing to the wrong set of reference sites. So this is kind of my view, and it's changed a bit after this morning's discussion, but as of last week, this was kind of my view on the day that I put this together. We're pretty good at defining if systems are healthy. The methods that we use, whether it be RivPax or our method, is good. And it's done in a quantitative manner. And in fact, what we're able to do now is to actually determine what type 1 and type 2 error rates, which I think is, how far do I want to go down that path? We're starting to develop methods of saying, if we assign a test site as disturbed, we can tell you the certainty with which we're correct. That is, are we saying it's disturbed when it's not? That's a type 1 error. And we're also coming out now with methods for determining if it is disturbed and we haven't detected it, how well are we doing that? That's the power or sensitivity of the estimate of the method, and those are what's called type 2 errors. So we're starting to quantify those now with these methods. In my view, and this is just my opinion, there's a great tendency, and I think this is a human condition, to be honest with you, to focus effort on new indices and new approaches. I think human beings just do that. We go for the latest fashion, whatever it is, and scientists are not immune to that human characteristic. And I don't know how many NABS papers you've been to. Well, I know you've been to a lot, Chuck. When the list of NABS papers, I've got a new index, new indices, here's my new index, but not tested in any rigorous manner at all. We need to move beyond just saying if a site is different to a reference. So I call that beyond pass and fail. And we talked about diagnostic capacity and stuff like that. So that's something we haven't successfully done yet. One approach is to come up with predictive models that include non-reference sites so that we can actually predict if a site is out of reference condition, what you have to change to bring it back into reference condition, or to develop scenarios where if we change a stressor by so much, well, you can change it by this much and it'll go out of reference condition. So those are sort of dirty models. This one is really important. We can do all the monitoring and assessment we want, but it has to be linked to both policy objectives and management objectives and sit within a framework where the information gets used from an assessment method. And I'm guilty as this as anybody of building what you think is a great method, but if it doesn't sit in that framework or that world, it won't be used. So this is, what I think. This is my list of where we maybe need to go next. The groups problem. And these are not priorized or anything like that. So if you remember, the first step in the way we build our predictive models at this time is to classify the biological community into groups. The idea being these groups represent different types of community assemblages. But this is the reality for us when we split sites into groups, and these are actual groups we created for a model for the Fraser River in British Columbia. You can see that these are not distinct groups. What we're doing is we're sampling, or we're quantifying a continuum of a biological gradient, often a single gradient, sometimes multiple gradients. And as you collect more and more sites in your reference database, which is what we tend to do, you tend to fill in that continuum more and more. So the groups are simply an artificial requirement of discriminant analysis, that the model building method that we use. So this is, when we first started, when you have a few reference sites, you tend to find this, because people tend to sample at the extreme ranges of the reference environment. And so your models work pretty well, because the groups are really distinct. So these are biological communities at reference sites. And as you get more sites, this happens, and your models tend to not work so well. And now, we're filling in the continuum. And these are real sites that I just abstracted, certain numbers of reference sites. The implications of this are that, so I've got 3 examples here where we've built models as we've accumulated more and more sites. As you increase the number of sites, the accuracy of your models in predicting sites to the correct group declines. So in the Yukon, we went from 55% to 48%. The Fraser, we went from 63 to 49. And in the UK, they went from 66 to 52%. I don't know what their latest model is. So that means basically half the time we're predicting a site to the wrong group, which doesn't sound very good when you're trying to convey this message to manager that we got a really good method. It's right half the time. Well, you know what the converse is, right? Now, what Richard and Ewan and Chuck would say to me is that, yes, but it doesn't matter for RivPACs because we use the probabilities of it belonging to all the sites, and that's correct. It does matter a lot for the methods that we use in Canada. Although, if you remember that slide of the Fraser River, of the real data, there is a lot of overlap among these groups. And that's where the model's having difficulty predicting to. And in fact, it's not so important that it is misidentifying because you have some appropriate reference sites anyway. So one thing that we did at the SFS that we had at one of our conferences that we had a special session to honor Richard. We actually tried to look at alternative approaches to modeling. And so we looked at a whole array of different modeling approaches. And let me skip by. So some of these actors are in the room. But basically, the approaches that we tried fell into one of four types. One was modifying discriminant function analysis. That's the method we use now with groups. So using a modified approach to that. The other was, instead of separating reference space into these groups, let's just try and develop a method that picks a subset of those reference sites that are most similar to the test site. So that's called nearest neighbor methods. Some regression approaches and methods, I don't have a clue what's going on, neural networks, and Fiona can talk about Bayesian because I still don't understand the bloody thing. tried some of these methods. And we used 3 data sets, one from the Yukon, one from the Great Lakes, and one from here, the ACT. and everybody used this method, built a model, see how it performed, and then tested it against a subset of reference sites that we'd removed, so we knew they were referenced. And those sites that we ourselves had artificially disturbed, so we knew they were disturbed. And we call those sympacted sites. So just look at the colors here, that's all you need to look at. And this is hot off the press, because these papers are still in review right now. So that we use the 2 standard current methods that are being used right now, the cabin method, which is the Canadian one, and RivPax/Osriva. Sorry, Sue, I didn't get to change it on that slide, because I was going to do that when I was in yesterday, and I was on a plane yesterday. So RivPax/Osriva, because Sue did that. So what I've done is indicated in yellow which performed better on which data set within the 2 standard methods. This is just in terms of model accuracy. And then in green, which of the alternative methods performed better or worse in red than the standard methods we're using. And that was just the internal performance of the model. Simon? Just how yeah, well, you'll notice I've only got I don't have information for some of the methods, and that's because they're nearest neighbor, and I don't know what the model performance is. This is just cross-validation accuracy. So how well it predicted sites to the groups. More importantly, was how well the sites performed in terms of assessing undisturbed reference sites and disturbed reference sites. So looking at it, don't worry about the numbers, they're just there for me, but so we have the method, cabin, Osriva, so on. We have the data set, and then how cabin performed, and because I'm Canadian-centric, I compared it to Canada. So again, the thing to do, the 4 columns that are important are none, low, medium, high. So none was a reference site, so that should be assessed as non-disturbed. The low, medium, and high should be assessed as disturbed. If the column is green, the method performed better than cabin. If it's red, it performed worse than cabin. So for the undisturbed sites, that is saying undisturbed sites are not disturbed, all the methods except the tiered DFA for the ACT data set performed better than cabin. Had lower type 1 errors in stat language. For the disturbed sites, however, the converse was true, and of course this is typical because type 1 and type 2 errors work together. Almost all the methods except CAN, which is a nearest neighbor method, performed worse, were unable to detect disturbance with at the low disturbance and the medium disturbance, and they were about 50-50 with the high disturbance. I can't say any more. So, that's where we're at in terms of looking at alternative measures. And if you look at the numbers, they might be green or red, but they're not that different. And the reality is, if a method doesn't provide much improvement, if it ain't broke, don't fix it. So we're at the stage now of determining where we need to go further with this. I think it would be fair to say this is probably one of the more rigorous tests of alternative methods that's been used. Simon, as an editor of the session, might disagree. So the next thing I'd like to talk about is taxonomic issue. We have a huge problem in Canada I mean, taxonomists, these are people who identify the bugs, are not being trained. The turnaround time on getting taxonomic samples identified is slow. It can take 6 months, a year. I have to confess, in my lab, I had samples that had been sitting on a bench for 2 years before we got to them to identify them. And I know what you're like, Chuck, but that was the reality for us. It's a slow process. So one of the new and emerging technologies that has potential is, so I just said that, is to use invertebrate barcoding. So this is where you, at its simplest level, you take your sample, you grind it up, you run it through a black box. Dave, you can maybe talk about it a bit more, but a black box which is identifying the DNA of all the animals in there, and it prints out a species list. In fact, I was at a meeting just a couple of days ago, and they're at the point now where you can actually just take a water sample and run the DNA through. And you know, visions of, you know, Dr. Spock with his tricorder, you know, being there, stick a probe in the stream, and your taxonomic list. There's a lot of issues with this, though. But I think this is probably the way of the future, so that they say 5 to 10 years, I say 15 to 20 years, this methodology will probably become the standard. It has been tested on specific groups. This is a paper by one of the guys who's done a lot of this work, Paul Hébert in Guelph in Canada. So with the mayflies, they were able to identify 69 of 70 taxa correctly. The way the approach is being developed right now, it tends to be done by taxonomic group, by taxonomic group, by taxonomic group. Because basically you have to build up a library of the DNA barcodes. Now for bioassessment, we need it for the entire spectrum of organisms. And so we need to do it on a geographic scale rather than the taxonomic sort of scale, if you like. And the costs are coming down, you know, in leaps and bounds. Figures of $10 per sample are now being sort of bandied about. Geographic ranges. In Australia, in Canada, just about everywhere except the UK, where they sampled the whole bloody country. But they got a little country, because it's about the size of the Fraser Basin there. But we tend to collect data sets geographically. So these are 4 separate data sets that were collected in the western portion of Canada. And the models work within each of these regions. But there are these areas in between which you think that the models could probably apply to. But you don't know, because you haven't sampled there. But the suspicion is that they could. And if you can build these larger spatial scale models, it does give you the potential for saving a lot of resources in building reference databases. So we did an initial study looking at the feasibility of this. And if you look at an ordination plot of the community, so these are the 4 base geographic areas that we looked at, you can see there's considerable overlap amongst the kinds of animals that you find across that huge latitudinal gradient from the US border up to the subarctic. The Yukon sites tend to fall out, so basically it's because there's less species rich and lower abundance at that end. But, you know, the Fraser, if you kind of just looked at the Fraser, the green triangles and the blue squares, you can see they overlay on each other pretty well. So that suggests that, in fact, building models at a larger spatial scale is feasible. Habitat predictors. So what is a predictive variable? It's, in essence, it's a numeric description of the habitat, of a habitat attribute such as stream flow, channel width. Well, they're selected because we know from research that they have a link with the ecology of the invertebrates. These are the things that drive the structure of communities. But for modeling purposes, we need to be sure that they are not modified by disturbance. Because you need to know, for example, probably the best example would be a power plant discharging warm water. So we know that communities respond to temperature. Temperature is an important driver for invertebrate communities. But if you use temperature and you're downstream of a power plant effluent, the temperature is going to be much higher, and therefore you'll be predicting a community for high temperature. And that's not what you want to know. You want to know what should be there without that temperature signal. So your predictors should not respond to disturbance. And then once you've got a subset of them, they are the ones that sort of predict what kind of assemblage of organisms you will have. So that's what predictive variables are. When we started in this business, those descriptors tended to be things that you measured at the site. So you'd go in and you know, take a water sample, you'd measure the substrate, you'd get the form of the stream, you'd measure the riparian conditions, so vegetation, those kinds of things. If you look at the first model that we built on the Fraser, all those were site scale variables, except for latitude, which you can get off a map. In 2012, the model now included, as well as site scale variables, landscape scale variables acquired through geographic information systems. So that's a major shift in what kinds of predictive variables are being used. So this illustrates it perhaps slightly differently. So in the 2000, the first Fraser model, the variables we had available to us, 5 of them were landscape, and they were derived off maps. 23 were some kind of measure of the channel and the surroundings of the channel. In the 2012 model, all the geology variables, all the land cover variables, all the climate variables, a lot more potential predictive variables were all acquired by GIS, as were most of the landscape scale measures. So drainage area size, perimeter, stream density. And again, the same issue with the Yukon. I think only over 80% of the variables are GIS-derived variables. And there are potential problems with this. There are advantages, but problems. The advantage is that the data, what I mean by consistency, that they're acquired from the same databases. So these would be national scale databases for various land cover or for various layers, is what they call them. It does allow you to use historic data sets, because typically data sets that were acquired were not acquired. There are big data sets from the Mackenzie River studies in the 80s and 70s. But habitat data wasn't collected along with them. By using GIS, we can go back and potentially use those historic data sets. And they are matched to the watershed scale of the site. So those are the good things. There are some major disadvantages with them that need to be addressed. It's not that they can't be resolved, but we seem to have charged, at least in Canada anyway, willy-nilly into using GIS without the same rigor of determining if they're appropriate or not. So our temperature data and rainfall data, we know these are important in the kind of assemblage that you collect, but they're 30-year averages. Well, what the hell has a 30-year average got to do with a bug that has a life cycle maximum of 3 years, probably of 1 year? So that's a problem. And the only reason we use the 30 years is because that's what's in the layer. There's a mismatch of spatial scale, potentially. The GIS for a small catchment of, say, 50 or 60 hectares, that land cover is, there's an intimate relationship with the channel. When you get to big rivers, they can have catchments of 5,000 or 6,000 hectares. Well, what the hell is, the vegetation that the upper reaches got to do with what the animal's seeing at a river site, could be 200 or 300 kilometers away. So there's that mismatch of scale that has to be addressed. And some of the folks at Griffith, I don't know if Fran is in the room, but she and her colleague Erin have looked at ways of confining the GIS collected data to the vicinity of the site, and those methods have a lot of promise. The link between what we measure as a habitat predictor and a GIS layer is not what the bugs see. So we're removing ourselves further and further from what the bugs experience. We tend to use everything that's available. We're a bit data sluts to some degree. If it's there, we'll use it. And that's not necessarily good. It's basically pragmatism. We'll try a whole bunch of variables, and if they give us a better model, largely we'll use them. So there's a bit of a disconnect between the ecology and the predictors that we use. As scientists and academics, we always want to be on the frontier of science and looking at the latest methods and the newest methods, because that's what gets funded. You can't publish a fifth paper and I built a new predictive model using, riv packs or cabin, that doesn't get published. Nothing new there. So scientists tend to be interested in developing new methods, a bit glommy on what's the latest fashion often because there's funding for it. At the other end of the scale, for any kind of national program, you need consistency in reporting. Because if I keep changing my model all the time, and it gives me a different result, and we just had this in the Yukon where the models that we use are built for placer mining, and in the latest model rebuild, more sites, different sets of predictive variables, a site that was identified as in band 2, so possibly disturbed, is now identified in band 1, undisturbed. And the manager goes, what the hell is going on here? So that tension between maintaining the same approach and getting the latest science in there, which you want to do, you want to keep your methods up to date, is one that's really quite difficult to manage. So all we do with the predictive models, as we have them right now, we tell you, is it in reference condition or is it not? But people need to know more than that. They need to know if it's not in reference condition, why isn't it in reference condition? So that's what we mean by diagnostic capacity. And there are an array of methods that potentially can do this. I talked about the dirty models, so where you include stressors in your predictive models. Simon did some work how many years ago, Simon? 12 years ago it was in essence a dirty model, right? Simon, pay attention. It was in essence a dirty model, Imperator. And I don't know why people really didn't latch onto it. And that would be interesting in itself. The models that we build now are just built with the taxa themselves, the identification of the bugs. But models can equally be built with functional attributes that in fact will start to tease out what is causing the change. And the other sort of approaches, the kind of expert systems that could be used by putting in information in a system that, well, if you got this bug and that bug, this is kind of what it means. Big rivers are a problem. And Ewan might talk about those in his talk later, but the one problem is sampling big rivers. Big rivers like the Fraser River, as it enters the Pacific, it's a huge river. So just there are physical barriers to getting in there. But the other problem is there often aren't reference sites in them. And basically, this is from the RivPac's website. I didn't plagiarize it because I acknowledged it. But basically, there is no method that can really deal with big rivers because most cities are on big rivers and they're out of reference conditions. So another approach has to be taken to dealing with big rivers. These predictive models, as they're formulated, cannot deal with assessment of big rivers. But this is another huge problem. This is from my perspective, so this is my little model of these three boxes of science, policy, and management. They should be, in an ideal world, be talking to each other and interacting and feeding back to each other. But, But, your talk, a couple of years ago, addressed this issue. And I don't think generally it happens. It doesn't happen that well. And as a scientist, this is my view that this massive amorphous policy thing, and we're the little guys at the end, and they're all talking down to us, but nobody's listening to what we say. And we tend to deal with the managers, not the policymakers. So that's, I think it's probably fair to say most scientists, that would be their worldview. I suspect that, and I don't know because I don't work in policy, I have the view that policy people and management people talk to each other all the time, or they think they do, and they think we're off here, and there's no connection with what they're doing because we're not giving them the information they want, and we're doing things that aren't relevant to them. I'm being a little facetious here, but there is an element of truth in there. I've been working in this business now for 40-something years and on a number of studies, and I've only got one good example of where the system worked well. And it worked well because the problem was clearly identified and addressed that it was an issue at the international level. Canada and the US had a Great Lakes Water Quality Agreement. That was the policy document that supported all the work. The issue in this one was contaminated sediments were impairing use because they had to be removed for navigational purposes. They couldn't remove them because the contaminants in them exceeded chemical guidelines. So they were at an impasse. So, you know, there was a whole array of discussions, and we were able to persuade them that if you used a biological approach, in fact, we could answer this question for you and give you a clear answer as to whether these sediments were contaminated, not just had high chemical concentrations, but were contaminated. They were having biological effects. It took us 15 years, and there was constant communication between the managers and us doing the science. It still took 15 years from the beginning of the program when we said, look, if we take the RIVPAX type of approach and apply it in the Great Lakes together with some other indicators, toxicity tests, we can in fact tell you if you have a sediment problem. We can tell you how much you need to change it to get back to reference condition. Because the chemistry at all these sites exceeded guidelines. The biology did not. The animals were living with this. Still took 15 years before they had a whole decision-making framework in place with 4 lines of evidence, what you did in terms of a management context when you got certain responses. But this, to me, is the ultimate big one. And this is the elephant in the corner. And this first slide is one that Stuart Bunn from Griffith gave me. These are the resources that are available in Australia. He presented this at ASL in Brisbane 2 or 3 years ago. A gradual increase in funding, and it's dropping catastrophically in Australia. And without resources, it doesn't matter where, it's very difficult to move forward on this issue. And in Canada, I would argue it's even worse because our government in Canada, with our Prime Minister, Stephen Harper, and I don't work for the government, and I can say what I like, is that government is totally opposed to science. They don't want information. They know what the problems are and what they need to do. For example, in Canada, they killed the long-form census. The census, that's where the data comes for future planning. They got rid of the long-form census. So many scientific institutes and institutions have been in decline. Scientists in Canada were even on the street protesting. I mean, this is something that just doesn't happen. I mean, you know, we're a quiet bunch. We're working away in our labs. They're so incensed about what's going on in Canada. So the climate for doing science in Canada, I think we've dropped our budgets for science, and it was small anyway in terms of GDP, by at least 100% in the last couple of years. So the resources, unless you're working in the oil sands, and there's one other issue they're working on, basically you don't get funded in Canada. And if you're a government scientist, you can't speak without having it pre-approved by the minister. I mean, there's a whole muzzling. So, and on that thrilling note, positive spin, I will end. The problem with those, generally, is the ones I'm aware of, which is, you know, there's the Chandler's and Trent Index and the ASPT in the UK and Europe. They have the Hills and Hoff Index in the US and Canada that we use. But almost all those indices, the scoring systems for the taxa are based on their response to organic pollution. So they work quite well for that, but if there's another kind of disturbance, they just, you know, they're not that responsive to it. I mean, it's one of, and they're used a lot still in the US, Chuck, I think, as part of a suite of metrics that, you know, that are available for description. But yeah, we're not that keen because they just focused on that one type of disturbance, which is, you know, an organic enrichment. Oxygen really is what they respond to, lack of oxygen. I think, yeah, the traction initially was because if you looked at what was before, it was pretty localized, pretty specialized. These were the first systems that were able to allow you to do large spatial scale assessments. But they're limited. they tell you if it's disturbance. And if you're going to be in a management context, well, okay, it's not in reference anymore. Now what do I do? And we haven't added that capacity to the standard methods that are being used. And I think that's one of the reasons. I think the other reason is, you know, the old argument that familiarity and contempt, you know, tends to play a role as well, but that's just a gut feeling. Yeah, I mean, people use it, but people, I think, and I said this earlier this afternoon, I think the expectation is it's going to deliver more than it delivers. It's very good at what it does, I would argue, in most cases. There are situations where big rivers is a problem, for example. Or the lack of a suitable reference sites is a problem. But over the broad sort of geographic context, certainly in my experience, is it works generally well, but it's the first step. And those extra pieces, you know, so it's very good at doing sort of trend analysis either through time or spatially, but it doesn't identify cause, and I think that's, you know, it's that deeper layer that, and I don't think you can blame the method for that. I think we need other methods, you know, to do those things. And that was always the intent with those rivers. It wasn't just supposed to be invertebrates. There were going to be more. There was going to be diatoms. There was going to be fish, but you know, I don't know what happened. I'm assuming it was a funding thing as much as anything, that the resources were never there to do that. The techniques that we used were what I call a tiered DFA. So instead of having one model that tries to characterize all the sites, all the groups at one time, was a series of models that we'd use that they certainly performed a lot better in terms of their accuracy in assigning sites. There were 2 or 3 nearest neighbor methods, so to pick out which were the nearest neighbors. Wayne, you did logistic regression, I think. It was what Wayne used, logistic regression, against each taxon. Yeah, so for every taxon, Wayne developed a regression model. And then there was Angus Webb in Melbourne used Bayesian, a Bayesian model. I think that was it. There were 2 or 3 nearest neighbor methods. I don't know. You know, the only knowledge that I have is they built a RivPax type model for Atlantic Canada using metrics rather than the taxa. So they used very simple things like richness, dominance, a couple of other things. And they quite nicely identified what the cause was, because I think it was a shift in dominance, and that was very indicative of what the problem was. I think the traits have potential, but the problem with traits, because you use the same data, you take the taxonomic list, and then you can generate a set of attributes for traits. The problem with traits is that, and there was one gentleman here who's kind of knows a lot more about it than I do, is, you know, the models right now, a lot of them are at family level, and traits at the family level are pretty insensitive, because you're dealing with a whole number of taxa that fit the different functional groups. So I think if you're going to go with traits, you need to go to genus level, and I'm not sure in Australia whether they go to genus level with the models, or with the identification. Sue? So some. In Canada, we do at reference sites, so we could build those kinds of functional models. That would be the cheapest alternative to look at first, because the data are already there to build those. Going to other kinds of things, you know, I talked about biomarkers. It wasn't that popularly received, but toxicity tests, that's a whole step up, because you now have to resample all the reference sites and build those responses.